Yeah. Second is efficient storage. Non-redundant data, sorry, redundant data takes up more space than necessary and can slow down the analysis process. By removing duplicates, the dataset becomes more compact and easier to work with. Next is better insights. Non-redundant datasets can provide better insights and more accurate conclusions. When multiple copies of the same data are present, it can skew the analysis and lead to the incorrect conclusion or biasness. Next thing is quality control. A non-redundant dataset can help to ensure the quality of the data. If there are duplicate entries, it can be difficult to identify the errors or inconsistencies in the data. By removing duplicates, it becomes easier to spot errors and ensure that the data is accurate or reliable. Next thing is improved generalization. So models trained on non-redundant data are less likely to memorize the patterns or features that are unique to the training set and can better capture the underlying patterns and relationships in the data. This also non-redundant using non-redundant data, we have chances of faster training in terms of machine learning models and others. Overall, using a non-redundant dataset is crucial for achieving better accuracy, efficiency, and generalization in the machine learning models. In summary, non-redundant datasets are important for improving the performance, storage, efficiency, quality control, and accuracy of insights in data analysis and machine learning. Okay, now we have so far we have understand what is the importance of one. Now we'll try to study what is the difference between redundant and non-redundant dataset with examples. Redundant dataset refers to the sets of data that contains duplicate or highly similar information. Redundancy in datasets can occur due to various reasons such as merging data from multiple sources or errors in dataset. Redundant datasets can be problematic as they increase the size of the data and make analysis more time consuming and potentially introduce errors if the duplicate data is not identified and removed. Whereas the non-redundant datasets, on the other hand, contain unique and distinct information without any duplications or unnecessary overlap. Non-redundant datasets are desirable for data analysis as they are more efficient, easier to manage, and less prone to errors. Let's talk about some examples. For example, a company has a customer database that includes name, addresses, purchase history. Then the same company has recently acquired another business with similar customer database. For example, we can only take a customer database of a particular restaurant, and when we go up to a particular database any particular shopping mall or any other restaurant, then we see some common people who are the customers of the both two. Then when these two databases are combined, then some customers are included in both databases, resulting in duplicate records. So this combined database where there are duplicate records is called as a redundant set. To make this dataset non-redundant, the company would need to identify and remove the duplicate records, so each customer appears only once. Coming to another example which I mentioned in the slide as well, a researcher will be studying effect of the drug on patients with particular medical condition. Then the researcher collects data from multiple hospitals, and some patients are included in the data sets from multiple hospitals. So this combined dataset is a redundant dataset. To make this dataset non-redundant, the researcher would need to identify and remove duplicate records of patient, so each patient appears only once. Next is how can we acquire a non-redundant dataset. So the popular algorithm is CD-HIT. So CD-HIT is a popular bioinformatic tool for clustering and comparing large set of protein or nucleotide sequences. It is a fast and efficient algorithm that can handle millions of sequences within reasonable time frame. CD-HIT can be used for various purposes including sequence clustering, sequence redundancy reduction, that is getting a non-redundant sequence dataset, sequence database construction and protein family construct classification. So we'll have what are the steps involved in. So the CD hit can be downloaded from the website http://cdhit.org, and the website provides installation instructions for various operating systems as well. Next step is to prepare your dataset. The input file should be in FASTA format with one sequence per record. In this process, the third step is to run CD hit using the following command, where input.fasta is the name of the input file, output.fasta is the name of the output file, and C flag specifies the cutoff or the sequence identity threshold. For example, here it's shown as 0.9, which specifies a sequence identity cutoff of 90%. That is, sequences with at least 90% identity will be clustered together. And the flag N specifies word size used for the alignment. That is, CD hit will compare sequences using 5 that is window size of 5. The next step is to interpret the results or analyze the output. The output FASTA file will contain clustered sequences with each cluster represented by single sequence. So this single sequence is called as a representative sequence. Think you have 100 sequence and you have to group them into the five groups. So each group equally. So each group will have 20 sequences, and one among the 20 sequences is taken as a representative dataset or sequence of that group. And this is very dynamic process, and the representative sequences will change when you rerun the CD hit on the similar dataset again. Here, the header of each representative sequence will contain the cluster information, including the number of sequences in the cluster and percentage identity between the representative sequence and the other sequence in the cluster. So this CD hit can also generate a file containing non-redundant sequences, that is a non-redundant dataset, taking one sequence per cluster, or it may take the representative sequence of that cluster. Overall, CD hit is a powerful tool for sequence clustering and redundancy reduction. It is widely used in bioinformatics research for various applications such as genome annotation, metagenomics, protein structure prediction. With its fast and efficient algorithm, CD hit can handle large datasets and provide valuable insights into the sequence homology and diversity. Okay, now we have understood what is the role. Now let's see how CD hit works. As I mentioned, the term CD hit refers to clustering and comparing large set of protein or nucleotide sequences. When we expand the word CD hit, the CD hit stands for Cluster Database at High Identity with Tolerance. It was first introduced in 2004 and since has undergone several updates and improvements. Briefly talking about, CD hit works by identifying the group of sequences that share high degree of sequence similarity and cluster them together into a single representative sequence. So this can be useful in many different applications, such as identifying homologous gene across the species. The widely used application reducing redundancy in large dataset, and also it helps in identifying the potential functional domains. Coming to the process, here are the main steps involved in CD hit clustering. Sequence processing. The input sequences which are in the FASTA format are first preprocessed to remove any low quality regions, such as regions with high level of noise or sequencing errors. This sequence may also be filtered to remove any duplicates or highly similar sequences that could bias the clustering results. Followed by that, sequence alignment. The sequences are then aligned to identify the regions of similarity and dissimilarity. CD hit uses an heuristic algorithm that first identifies short words shared between the two or more sequences and then attempts to extend these words into a longer alignments. That is, start with a 5 and search for their repeats in the other sequences, then attempts to extend this by extend these short sequences in the alignment to generate a longer alignments. The next step is clustering. CD-HIT uses a clustering algorithm to group together the sequences that share high degree of similarity. So in the sequence alignment step, if two or more sequences are termed to be having a cutoff or similarity identity that is equal to or more than the threshold, then they are clustered together. So in this clustering step, the algorithm first identifies the seed sequences that are highly representative of the given cluster and then adds additional sequence to the cluster based on their similarity to the seed sequence. Seed sequence could be the representative sequence as well. The clustering process continues until all the sequences have been assigned to a cluster. Next is cluster filtering. The CD-HIT allows for different levels of cluster filtering based on the sequence identity threshold. For example, you can only choose to keep the clusters with certain level of sequence identity and discard any clusters with lower identity. This can help to reduce redundancy in dataset and make downstream analysis more efficient. Finally, how the output looks. The final output is a set of representative sequence for each cluster along with information on cluster size and sequence identity. The output can be saved in various formats such as FASTA to be further used in downput analysis. Okay, since we have talked about clustering in CD-HIT, let's talk about the famous clustering algorithm, that is K-means algorithm, which is used to partition a set of data into K clusters based on their similarity. The algorithm, this K-means clustering algorithm, works by iteratively assigning the database to the nearest cluster center and then recalculating the cluster center based on the new assignments. The process continues until convergence, that is, until the cluster assignments has no longer change. So since we are talking about the cluster and other clusters, how do we differentiate which member to be in the cluster and which member not to be in the cluster? So we build clusters such a way that within the cluster all the members have similar or have high percentage identity, and when we take among the clusters there should be the less sequence similarity or identity. That is, within the cluster all are same, among the clusters no one is same. That's how these clusters are built, and this process is an iterative process. This goes on until no change of clusters is observed. So the steps involved in K-mean clustering algorithm are: the first step is choose number of clusters that we have to build. That means number of representative sequence that we require. So this step requires some prior knowledge or intuition about the data. So you should have an idea of how many distinct groups you want to identify. For example, when we take a class of students, so how we differentiate them? So we give the k size as 2, that is boys and girls. So for this, deciding the k value, we need a prior knowledge on the data set or data that we are going to work with. Second step is randomly initialize k cluster centers. That is, randomly select, okay, this sequence is the representative of cluster 1, or this sequence could be for 2, this could random assignments. Next step is assigning each data to the nearest cluster center, then based on the distance. Now once the cluster centers are assigned, the initialized, then we take each data point and check to which cluster this data point is highly similar or highly identical, and then we assign this data point or sequence into that cluster. So for example, Euclidean distance, Hamming distance, that is number of changes in the sequence, number of mutations happen. So identity score, similarity score were used for this assigning purpose. So once this assignment has been done, then there is a process called recalculating centroid of each cluster based on the data points assigned. So this means that the new centroid becomes the cluster center now. So the steps 3 and 4 are repeated until the cluster alignment has no longer changed or a maximum number of iterations has been reached. In some cases, when we are working with the closely related sequence or the proteins within same organism, there is chance that the algorithm will never converge. So in that case, we use a cutoff iteration size. So after this number of iterations, the algorithm will stop by itself. So once k-clustering has been converged, the resulting clusters can be analyzed to gain insights into the data. So the next is other widely used application PISES. So this is a software tool used for the curation and filtering of protein sequences in bioinformatics. It is primarily used for creation of high quality reference datasets for use in sequence similarity searches, protein structure prediction, and other applications in the field of computational biology. So this tool works by taking a set of protein sequences and filtering them filtering so this takes and filtering them based on the variety of criteria such as sequence identity, length, and presence of non-standard amino acids or caps. The resulting dataset is then clustered using an hierarchical cluster algorithm which groups similar sequences together based on their pairwise sequence similarity. The clustered data is then used to generate a non-redundant reference dataset which could contain a representative set of sequences from each cluster. This reference dataset can be used for a variety of applications such as sequence database searching, homology modeling, and protein structure prediction. One of the key features of this tool is its ability to filter sequences based on their structure as well as their sequence similarity. This helps to ensure that the resulting reference dataset is of high quality and suitable for use in a wide range of computational biology applications. Overall, this is a powerful tool for curation and filtering protein sequences and has become an important component in many bioinformatics. So far, we have learned two tools that are similar. So let's understand what is their difference. Yeah, CD-HIT uses a fast and memory efficient algorithm to cluster the sequences based on the sequence identity, and it can handle millions of sequences with relatively low computational requirements. Yeah, can you have seeing a disturbance? Can you please unmute or mute yourself? Sorry. Whereas PISES applies a series of filters to remove the sequences that are considered low quality or potentially error, and also provides options for controlling the sequence identity threshold and other filtering criteria such as sequence length and structural quantity. Next, let's start with the protein structure, secondary structure. Proteins are complex molecules that composed of long chain of amino acids. The way this long chain of amino acids are arranged in space determines the overall protein space. And specific shape of the different regions of the protein are referred to as, or the regular arrangements of these protein are called as secondary structures. There are three main types of secondary structures: alpha helices, beta sheets, and loops. Alpha helices are tightly coiled structures that resembles a spring. They are formed by a single polynucleotide polypeptide chain that coils around itself, forming a right-handed helix. The backbone of the polypeptide chain forms the axis of the helix, and the side chain of the amino acids point outwards. The alpha helices are stabilized by hydrogen bonds between the amino acid backbone and are often found in core of protein. Next is beta sheets. Beta sheets are flat and sheet-like structures composed of multiple strands of polypeptide chains that are arranged side by side. The side chain of the amino acids that point alternatively up and down from the sheet, and these sheets can be either parallel or anti-parallel. Beta sheets are often found on the surface of the proteins and are important for providing structural stability. Next is loops. These are also known as turns or coils, which are the regions of polypeptide chain that do not form regular alpha helices or beta sheets. They are often found on the surface of the proteins and are flexible, allowing the protein to interact with the other molecules or undergo conformational changes. Loops are held together by various types of bonds and interactions, including hydrogen bonds, van der Waals forces, and hydrophobic interactions. Overall, these regular secondary structures of the protein are important for maintaining the three-dimensional structure of the protein and play a critical role in protein function. Understanding the structure of the protein and the secondary structures is essential for understanding their biological functions and designing the drugs and other therapies that target them. And there is an important concept of what are dihedral angles in proteins. The dihedral angles in protein refers to the angles between adjacent chemical bonds in the peptide backbone. Specifically, the dihedral angles are the angles between plane defined by the nitrogen atom, C alpha, and carbonyl atom of one amino acid residue, and the plane defined by the C alpha, carbonyl carbon, and nitrogen atom of the next amino acid. So this is the angle defined by the two planes, each having three atoms, each plane having the three atoms from the two different amino acids. So these dihedral angles determine the conformation of the protein backbone and play a crucial role in protein folding and function. So there are two main types of dihedral angles in the protein backbone. There are phi and psi. The phi angle is the angle between the nitrogen C alpha in the peptide backbone, whereas the phi is the angle between C alpha and the carbonyl atoms. The values of these angles are often plotted in the Ramachandran plot, which shows the distribution of phi psi angles in the protein. That is, what are the allowed combinations of phi psi in protein structure. So the Ramachandran plot is a useful tool for analyzing protein structure and identifying the regions of the structure that may be energetically unfavorable. Next is the DSSP algorithm in bioinformatics. The DSSP algorithm stands for Dictionary of Secondary Structure of Proteins, which is a widely used program in bioinformatics for analyzing the secondary structure of proteins. It was first introduced by Cab and Sander in 1983 and has become an essential tool for researchers studying protein structure and function. The DSSP program works by analyzing the coordinates of each amino acid in a protein structure, which can be obtained from amino acid nothing but the PDB file or the three-dimensional coordinates, XYZ Cartesian coordinates of the amino acids and their positions. So these data can be obtained from X-ray or NMR experiments. So this DSSP program assigns the secondary structure state to each residue based on its position and orientation of its backbone atoms. The secondary structure states are classified into 8 categories, that is alpha helix, 3-turn helix, 5-turn helix, pi helix, extended strand, beta bridge, bend, and random coil. That this program also assigns an hydrogen bond state to each residue, which can also be used to analyze the stability of the protein structure. Finally, the DSSP program generates an output file that contains the summary of the secondary structure, and the hydrogen bonding pattern of the protein, as well as detailed description of each residue position and their orientation in the structure. This information can be used to study the folding of the protein protein-protein interactions and effect of the mutations on protein structure. The secondary structure information generated by DSSP is used to annotate the PDB files, making it easier for the researchers to identify the structural motifs and compare protein structures across the different species. Overall, DSSP is a powerful tool for studying the protein structure and function, and its widespread use in scientific community has helped to advance our understanding of biological systems at molecular level. Okay, now we have studied one method, DSSP. Let's study the generalized methods that help in predicting the protein structure. Protein secondary structure prediction. There are several methods for predicting the protein secondary structures. That is nothing but which refers to the local conformational arrangement of protein backbone. So broadly, these can be characterized into experimental and computational methods. So experimentally, they are the NMR, X-ray, so on. Coming to the computational methods, we have starting with the sequence-based prediction methods, where the methods involves using algorithms to predict the secondary structure of a protein based solely on its amino acid sequence. So where the prediction is done by using statistical methods or machine learning algorithms. Next is the homology-based prediction method, which involves comparing the amino acid sequence of a protein to a database of known protein structures. Can anyone name in the chat box what is the database of known protein structures? Yes, thank you for the answer. Yeah, it's PDB. The third is energy-based prediction, where this method involves the predicting the most stable conformation of a protein by calculating the energy of each possible secondary structure using molecular. Next is the hybrid method, which is the combination of what I discussed above. Here, we'll be discussing sequence-based methods detailedly. The common sequence-based methods are Chou-Fasman, use of neural networks, hidden Markov models, support vector machines, and evolutionary conservation method. So each of these I'll be discussing separately as well. So let's start with the Chou-Fasman method. The Chou-Fasman method is a well-known algorithm for predicting the protein secondary structures, which uses amino acid sequence of a protein to determine the probability of each residue being in an alpha helix. That means it uses amino acid sequence and then determines the probability of each residue that is part of the sequence to be in an alpha helix, beta, or a random coil. So this method was developed by Peter Chou and Gerald Fasman in 1970 and is based on observations of certain amino acids that tend to occur more frequently in secondary structures. This method assigns the score to each amino acids based on this propensity to form a particular secondary structure. For example, if we take an alanine, there is a pre-calculated value using the existing structures, that is propensity of alanine to be in alpha helix, propensity of alanine to be in beta sheet, and propensity of alanine to be in some random coil. So these scores are then used to calculate a probability that the given residue is in a particular secondary structure. These scores are nothing but probability values corresponding to their observance in particular secondary structure. So this method involves calculating a set of parameters for each amino acid based on their propensity to form different secondary structures. These parameters are then used to calculate the probability of each amino acid being in particular secondary structure. Next thing is information theory or GR method. So information theory is a mathematical framework that can be used to measure the amount of uncertainty in the system. So in the context of protein secondary structure prediction, information theory can be used to measure the amount of uncertainty in the relationship between the amino acid sequence and secondary structure of a protein. One common approach is to use a probabilistic model to estimate the likelihood of each amino acid being a part of secondary structure element based on its surrounding sequence, that is combined neighborhood analysis. The most commonly used probabilistic model for protein structure prediction is hidden macro model. Next thing is uh, GR model. So this model is another popular method for protein structure prediction. So this is a machine learning based approach that uses a sliding window to analyze the sequence and predict the secondary structure based on the surrounding amino acids. So this method uses a scoring matrix to assign a score to each possible secondary structure element based on the amino acid sequence in the window. The element with highest score is then assigned as the predictor secondary structure for that position in the sequence. Next thing is hydrophobicity profile. So these are important because they allow for the identification of a region of a protein that are likely to form secondary structures based on their relative hydrophobicity. Hydrophobicity refers to the tendency of a molecule or a portion of a molecule to avoid contact with water, and this property can be used to predict the following folding patterns. For example, regions of high hydrophobicity are often associated with alpha helices, while the region of low hydrophobicity are more likely to form beta sheets. So till here, the discussion part has been done. From here onwards, we'll move to the sample problems. So the first question is, in cd hit command, what does the flag N stands for? You can answer the question using the chat box. Yeah. I request everyone to turn off their videos so that others who are having the lower bandwidth can access the without any interruption. Yeah, I started receiving some answers. That's good. Yes. I hope my slides are visible and everybody can see the first question. If someone cannot see the first question, they can raise their hand. I'll help to solve their issue. Everyone can see if someone is unable to see the screen, use hand raise option or let me know through the chat box. Okay. Yeah, as everyone mentioned, answer D is correct. Okay, let's understand some of the important flags. The I flag stands for the input flag. So after the I, we give the input file containing the sequences that used to be clustered. So these sequences can be in FASTA or FASTQ format. O stands for output flag. So this, whatever the name we give, the output is written into that. Next is cutoff. This cutoff is also called as the sequence identity threshold for clustering. So sequences with an pairwise sequence identity greater than or equal to this cutoff will be grouped together in the same cluster. So the default value for the CD hit is 0.9, but this can be adjusted by depending on the desired level or clustering string. Next is N string. That is what the question is. Is the word used to compare the sequences. That is, we have mentioned about the seed sequence where it compares and then it starts building on. That is this. The D is the max memory that is allowed to use. There are some other that is M, which is cluster method. Then is T, that is number of threads for the parallel processing and all. It is too much computational knowledge. Yeah, next question is, Given equations of two planes, can you calculate the dihedral angle of this? Or even you can calculate what is the cos theta of it. No need to do the inverse. I think it's a bit of, you need the inverse table and all. So given equation of two planes, or when you have the two vectors, how will you calculate a dihedral angles that is formed by these two planes? I know this is a stringent calculation routine. I'll wait for the time that you people calculate. So generally, the dihedral angle is the angle between the two intersecting planes. To calculate the dihedral angle using the equation of the planes, we can use the dot product between the normal vectors of the plane. So once you get normal vectors from the equation, do their dot product, you get that. Okay, given these two are the equations of the plane, now next step is to find the normal vectors. So for plane A, the coefficients of x, y and z will be the normal vector. That is 1, 7 and minus 3 are the normal vectors for plane A. And similarly for plane B, 3, minus 2 and 4 will be the normal vectors. Now we have the normal vectors, then we have to calculate their dot product. Can anyone tell me what is the dot product of these two vectors? So dot product is nothing but summation of the product of x coordinates, y coordinates and the z coordinates. So here, okay, sorry. So here the dot product will be 1 into 3 plus 7 into minus 2 plus 3 into minus 4. So it comes minus 23. And similarly, the formula for cos theta is dot product multiplied by magnitude of each vector. Magnitude is nothing but square root of the sum of the squares of the each vector value. That is, when you consider the first vector, we have 1, 7 and minus 3. So the magnitude value will be 1 square plus 7 square plus minus 3 square under root. So that is under root 59 square. 59. Similarly for B, the magnitude will be 29. And when we apply, the cos theta value is given as this. And the final answer is 126.37. Okay, next is a similar question, or try to answer this question. Which of the following conditions best represent a set of non-redundant sequences? I'm repeating the question again. Which of the following conditions best represent a set of non-redundant sequences? That is, no two sequences have a similarity of less than 90%. No two sequences have a similarity of greater than 30%. No two sequences have a similarity greater than 90%. Or no two sequences have a similarity less than 30%. Yeah, please go ahead. Someone has raised the hand. Okay, I've seen some answers. So the final aim of a non-redundant sequence is to have a set of sequences that has very less similarity among them. Okay, I have seen two different answers. Okay, let me move forward to the solution. The solution is B. As I mentioned, the solution B states no two sequences have similarity greater than 30. So the possible values of similarity size which are less than 30. And when we check the D option, no two sequences of similarity less than 30. That means here the option comes 40, 50, 60, which is against the similarity rule. Okay, here is the basic question. What does the K stand in K-means algorithm? What does K stand in K-means algorithm? I think this is pretty straightforward question. Yes, I got an answer already. Yeah, you guys are free to answer whatever you feel. It's just an learning experience for all of us, including me here. Yeah, that's good. One more answer. Yeah. Okay. The answer is number of clusters. Okay, let's stand. The given option B is number of data. So if number of K means algorithm, so K ways of doing it, or K output has to come from it. So here the letter K stands for number of clusters that algorithm should create, and this is considered as an hyperparameter that user specifies when applying the algorithm to a particular data set. For example, if the user specifies k equals to 3, the algorithm will try to find 3 clusters in the dataset, with each cluster containing the data points that are similar to each other. So this value of k can have a significant impact on the quality of resulting clustering. If k is too small, the algorithm may group together data points that are not actually similar as well, resulting in poor clustering. If k is too large, the algorithm may split similar data points into separate clusters, resulting in overfitting and poor generation to new data. Therefore, it is important to choose an appropriate value of k based on the characteristics of data being analyzed and the goals of the analysis. Okay, next the question is based on the dihedral angles that we discussed. The 5-side torsional angles are rotation along what type of bonds respectively? Again, two different answers. That's good. Yeah, let's learn them detailedly. The answer is B. But as we all know, psi and phi are the two angles of the main backbone torsional angles in proteins, which describe the rotation around the peptide bond. So the two possible bonds that we could see around the peptide bond is NC alpha. You can see here NC alpha and C alpha C beta. So the phi angle is defined as the rotation angle around the bond between nitrogen and C alpha. So NC alpha bond rotation corresponds to the phi angle, and the psi angle is defined as the rotation angle around the bond between alpha carbon of the first amino acid of peptide bond and the carbon atom of the second amino acid in the peptide bond. And these values are often represented in Ramachandran plot. I hope that should be the next question. So in Ramachandran plot, the allowed regions of alpha helix conformations are restricted to which quadrants? Okay, let's look into the answer. Answer is C. So actually the possible values for dihedrals are minus 180 to plus 180. So on x-axis we are having phi value, on y-axis we are having the psi value. This is actually the Ramachandran plot. So the next question is, which of the following are known as helix breakers? The answer is pretty straightforward. Proline and glycine, because proline that introduces a kink in the protein backbone due to its cyclic structure, so that can disrupt. And glycine being the smallest amino acid, and its side chain is a just single hydrogen atom. So this allows for high degree of flexibility in protein backbone, which can also disrupt the alpha helix structure. So this is the final question. PhD in secondary structure prediction method is based on. Consider this is a kind of hybrid method. I didn't explain about it. Sir must have explained in the class. Yeah. And I think this is the last question for the day. If you have any questions, please feel free to write or ask through the mic, write in the chat box, or you can unmute and ask yourself. If no question, thank you for attending. You are free to leave. The answer is multiple sequence alignment and neural. I mentioned hybrid methods are combination of several individual methods that are previously discovered. That's all for today. Question number 2. 1 second. See, in question number 2, these are the two plane equations that have given. So the first step is to extract the normal vectors. Normal vectors are nothing but the coefficients of the x, y, z. So x has 1, y has 7, and 3 has, z has -3. So this is the normal vector to the plane A. Similarly, normal vector to plane B will be 3, -2, and 4. So once we have the normal vectors, the next step is to find the dot product of normal vector. Dot product is nothing but x value, multiply x value plus multiplication of y values plus multiplication of z values. So here we will be having 1 into 3 plus 7 into -2 plus -3 into 4, which is nothing but 23. And when we come with the cos theta is dot product divided by magnitude of each vector. Here magnitude is nothing but sum of the square root of the sum of the squares of the each element, so each value in the normal plane in simpler terms. So when we consider the normal plane 1, we have 1, that is 1 square, we have 7, plus 7 square, we have -3, plus -3 square. So 1 plus 49 plus 9 will give to square root 59. That is, this is sum of the squares. So square root of 59 is the magnitude. Similarly, 3 square is 9 plus 2 square is 4, like -2 is also 4, and 4 square is 16. So you will get square root of 29. So the cos theta value here, cos theta is the trigonometric value where theta is the angle between two planes. Is this? Overall, to find the theta value, we use the cos inverse. So it is 126.37 degrees. Hope I'm clear here. Yeah, please be fast in asking the questions.